We start the session with part of our workshop. And Nandan is going to do for us. He's military fellow at KPH in EuroCC for 4 years now, I think.
3.5, yeah.
Okay. He's been conducting great work. I just looked it up, and before that he was with among.
Task Force Shield.
Okay, so I can never pronounce his last name, but he was at where NEMT was born. Now he's with the GROMACS group, so he knows all sides of the story, and he will be talking to us about molecular dynamics simulation with the hands-on session later on. Yep. Thank you very much for being here.
Yep. Thank you so much for the kind introduction. So I'm going to talk about GROMACS simulations, and so I thought my voice might be a little bit down because I was sick last week, so hopefully I will be able to speak properly. But I'm going to do a bit of more interactive. I'm going to have some small groups discussion and then kind of get some questions here rather than giving me lectures for the full hours. I feel like that way can be. I also want to know you more. What's your backgrounds are? Okay. So as already, I think I just presented myself about, I did my bachelor in India, and then I went to the US for my PhD, and now I'm in Stockholm doing postdoc. And in the Stockholm team, this is what we are. And this is Eric Lindahl. Yeah, I think you can, yeah, maybe you can use this thing. Yeah, that's our PI who does a lot of GROMACS management and development. And we do both, not just development of GROMACS, we also have applications. For example, I'm primarily come from using simulations to apply new biological problems. And also not just simulation, also we use a lot of machine learning based approaches and also try to integrate them with different experimental data, for example, cryo-EM or small angle scattering data. But today I'm gonna mainly focus on the simulation part. In Stockholm, the summer and winter could be quite different. Summer is quite nice and beautiful and sunny, but winter could be quite horrible sometimes. But we have very nice lights in the night in the winter, which you can see, which you cannot, I think, only see like on the northern region. So you're welcome to come both in summer and winter to see the beauty of different sites. Okay, so I'm going to actually start with. Before that, actually, what I'm going to do, I'm going to ask these questions and divide in smaller groups. And then I'm going to use the menti.com, if I might see menti.com, which you can scan the QR code. And then you can respond to this one where I'm going to show you right now. Okay, I think I have to go again. Okay, so once you can scan the QR code here, and then you can, but before doing that, first, I would like to have you in a smaller group. For example, let's say like 4 or 5 people can hear, another few people here, and then kind of gather. And then I'm gonna give you 5 minutes to have this question in your mind. Why do we do simulations in the first place? Because we could have not done simulations, just let's say do experiments, right? Why would we have to do simulations in the first place? Maybe you're gonna give some thought about this, and then you can respond by using this QR code, or just go to menti.com and hit this code right here, and then you can respond here by typing, and then we can have some discussion from your side and then answering questions, okay? Yes, so I would suggest maybe turn down your chair so that you can be more interactive. But you can do the same thing also during the practical session as well, so that you can be more working together. Otherwise, it could be quite boring to work by yourself.
Maybe in pairs, because it's so easy to get in pairs.
Sorry?
In pairs, maybe.
Yeah, sure. Yeah, in pairs, yeah. You should talk. It's fine to make it loud and noisy. It's fine. Just keep talking.
Allah ben para gideceğim.
I would have to do it Zoom people though. They can also see, so they can also respond there as well. Sorry. I think Zoom in Zoom also they can also see and then they can respond as well.
They can see.
Yeah.
Here.
Yeah. Yeah. Perfect. Yeah. Yeah.
Laptop'tan.
Nasıl?
Menti.com diyorsunuz. Koruyoruz.
All right. It seems like there are a lot of response already, so we can. One of the most thing comes is docking, which seems to be the most highlighted one. Yeah, docking is. Yeah, you can. Maybe we want to respond. Whoever wants to respond about docking. Anyone wants to respond?
Understand molecular interactions.
Yeah. Oh, in that sense, yes, exactly. Yeah. 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 Docking in a more traditional sense. Sorry. Yeah. 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 And I see also see to understand dynamic. I think this would be. Oh, it doesn't work here. I think. Right? Yeah. This also. Yes. Yes. Understand the dynamics. That's one of the extremely, I think, essential elements of simulations, which we try to understand. Not just a static structure. For example, you can get this from. There's a cryo-EM, right? You might have a cryo-EM or X-ray crystallography, where experimentally you can solve the structures using in the wet lab. But normally the problem is because we're studying dynamics, because these molecules are really, really small. And studying these small molecules is very, very precise time scale, for example, nanoscale, nanoseconds or microseconds is quite difficult. So that's the idea of why one of the most strong point of using molecular dynamics simulations. And predicting, of course, we want to understand new information. It's not just about having a protein and simulate and then see what goes on, right? You also understand new biology, new questions, which I'm going to actually give one example of from my own research, what we can learn. That's one example, but there are a lot of different examples, of course. Let's see, before going, and maybe. Does anyone to say something before we move on? All right. Okay, so I have to switch back and forth because I'm not sharing. I think I can just share the whole thing, I think, right? This will be easier, I think. Okay, I'm going to share the whole thing. Okay, I think you're going to see the answers. But so what do you see in this picture? What is the one word come to your mind? You might have seen the answer already, but. Yeah, sure. So poison, right? This is one of the molecule which interact in our different regions, but one of the most uh, major target is in our brain, in our neurons. And neurons are, for example, if you might have taken the most like preliminary biology course where you know that like when our signals transmit in our neurons, normally these neurotransmitters release in this synaptic cleft, and then it talk to these molecules called ligand-gated ion channels. These are the proteins which receive these neurotransmitters and then open up for ions to flow through. And that's how the signals in our brain can propagate. And this is one of the signals which can be interfere with a lot of different drugs. And it's important because, as you can see here, you can also see this in electrophysiological setup in experiments. For example, once these molecules binds, and then you can see a fire of currents, which goes through. And then it goes to this another state called desensitized state, even though it's not same as this one because the ligand is still bound here, but it's still closed state. So it very transiently open, and it goes to this closed state. And one of the project, yeah, there are different drugs, for example, cannabis, anesthesia, nicotine, a lot of different drugs binds here. And one of the projects which I was working on, which recently published, is that this open state of this channel was still missing in terms of how it's structured, it looks like. And the reason, which I already mentioned partially, is that it's very transient. So it opens up in a microseconds regime, which is a very precise or very small time scale to capture in experiments. Because let's say if you do experiments, if you wanna put the samples, then if you wanna take picture of it experimentally, this having the picture in the microsecond regimes or microsecond resolution is quite difficult. It's very small time scale. So that's where these experiments normally struggle. And that's where one of the ways to tackle this problem is using simulations. Because in simulations, in particular in molecular dynamic simulations, we try to preserve all the atomic details at the microsecond, we simulate this in the femtosecond regimes, and then we can resolve up to nanosecond, microseconds, even to millisecond regimes. So that's the power of simulations comes into play. So this was the question mark here, which I'm showing here. It doesn't work anyway. Yeah. So this was the question mark. We know how the desensitized or the closed state looks like, but we don't know how the open state looks like. And if you can also think about this in terms of free energy. What I mean by free energy is that, well, desensitized state can be captured because it's much more stable, which means that in the energy-wise, it's much more lower energy than for example, the one, which is the open state, which is high rate energy. And so that's the question, and this is the one which, yeah, I was trying to tackle this problem with simulations and see how the open state might look like. And I'm going to go to a bit more details. Taking the example, I'm going to go in more details what else we can learn from doing this kind of simulation. Okay. So now the second question comes is that the way I presented is that, okay, simulations are really cool. You can do everything, right? Which experiment cannot do. But you might already guess that this is not the case. As always, with any techniques, there are always drawbacks of simulations. And I think it's very important to, before doing simulation, it's important to know what is the strength and what is the weakness of this kind of method so that we know what problems we're tackling, or should we even try this problem at the first place with this particular method or not. It's very important to know so that we don't, let's say, waste our time or something like that. Okay, so this question again, I'm gonna do the same thing. I'm gonna go to this one, and then I'm gonna do next slide. And you should be able to see in your same thing, menti.com, should be able to respond to this again. And you're allowed to talk, of course. Okay, I think so. The most so far, the reliability is one of the most thing popping out. Anyone would like to comment on reliability? I think it's extremely important elements of it. Force field, yes. Do you want to say more about force field?
The force fields are limited. For example, you can use the GROMACS force fields for the amino acids, but if you have some complex molecules, you cannot directly use them. You have to do the political parameterization. So it's a difficult task.
Yeah. Yeah. No, that's really good. Good.
And also time scale.
Yeah, like when I elaborate on the time scale problem as well. Do you want to elaborate on the time scale problem as well? Is that's what you use. Yeah.
Well, and then something.
Yeah. Yes, exactly. Yeah. Anyone else? Let's see. Computational cost, yeah, is related. Expensive related, cost related. Size limit. Do you want to say anything about size limit? It's a small one, but I think it's a really important one as well.
It's related to the structure of the protein. The protein is very huge. We didn't do simulation accurately. But of this, we are using more the protein that has in range size, not the usual protein we couldn't do simulation.
It's gonna be expensive, yeah.
More parameters and to scoring to determine just to give us the exact score or like the result that we are looking for.
Yeah, so I think there are two elements of it, right? One is the two major elements. One is the force field, because in atomistic molecular dynamics simulation, normally we use classical approach, which means that we don't use any quantum effect explicitly. Of course, we try to sort of mimic this with parameterizations, and but these are not nowhere near perfect. But of course, models could be wrong, could be right. So that's why it's very important to understand the limits of what we can do. And as you explained, if you're working with a novel molecule like new drugs, then of course no one has done this before parameters. Normally for proteins it's fine because proteins are just 20 amino acids, right? They're parameterized heavily normally. But small molecules, drug molecules, new drugs, these are not. You have to do it yourself. Then you have to take care of all these different, like how you take care of the quantum effect, everything like this. So there's a whole new field to this, and it's quite complicated in general. Yes. And in terms of time scale, there's another drawback, right? So we use classical laws of motions like Newtonian dynamics. And the time integration we do is at femtosecond regimes, which is 10 to the power minus 15, right? Yeah. Picosecond is 12, yeah, sorry. Yeah, 10 to the minus 15, that's the time scale resolutions we do. And so then you have to do this integration at this very precise time scale because we care about the precision of our simulations. But the problem becomes that going up to this ladder, it becomes a problem. For example, if you use the PDC is the one we use in Sweden in KTH, but let's say you use the supercomputer in Turkey, then you might get, if you run for a couple of weeks in a reasonable system, so let's say proteins which is not too big maybe, then you might get regime of microseconds. Then if you throw GPUs on top of it, maybe you're gonna get a couple of times more faster. And if one of the very high level hardware, which is called Anton, which you might heard about, this is in the US. And this person is very rich and famous, Wall Street money, basically, to build this machine. And if you use this kind of very high-purpose architecture hardware, then you might get to the millisecond regimes where you might see like meaningful things happening, because we care about not just things wiggling around. We want to see meaningful things happening. For example, we want to see how a protein conformation change happens, goes to, let's say, opening or close, or how ligand and protein bind and unbind. And also we care about statistics, which means that we want to see these things just happening once or twice. We want to see this happening multiple times because we care about the free energy calculations. We care about the kinetic parameter calculations. So these are all matters that we sample things exhaustively so that we get reliable number, which is not really feasible with just launching simulations. That's not very practical. That's why you need, as you mentioned, that you need enhanced sampling simulations. That's one of the things we need. But we're not going to cover enhanced sampling in this workshop because we plan it to be more the basic GROMACS tutorial. So, but I'm not going to go too much details on the enhanced sampling part today. But we can, you can ask questions to me if you have any regarding this, okay? And this also brings this problem with this gap between experiments and simulations, because simulations are good at this smaller time, smaller length. Experiments are good at typically for larger time, larger length. And there's always problem of this, how do you fill this gap? Because we are not talk to each other. Computationalists and experimentalists wants to talk to each other because that's where the interesting thing happens. But so that's why there's a lot of completely new field of how do you do the integrative modeling, for example, as you heard today about you don't maybe rely on completely on simulation, maybe you want to do something else, take some restraints from these experiments and try to integrate in simulations or docking or anything like this. So there's a yeah, it's a completely different field how you want to do this more as reliably or as tightly as possible. Okay, and also this is also not we're gonna go through for the workshop. Also, we're not gonna go through this one today. All right, so the third one, which we have, is then, of course, now there is one problem is force field. That is a different topic, but let's talk about the time scale. Actually, yeah, you already have mentioned about the enhanced sampling, so maybe we don't need to, but so yeah, let's not take this question for now because I think we have sort of discussed already. But what I'm gonna show is that how we did it, or at least I did it in my case. I'm going to show you one quick example. I wasn't planning to show this, but I'm going to let me unskip this one and then let me just quickly go through this. Of course, just bear in mind that this is not the only way to do it. There will be multiple different ways to do it, but this is one of the ways to do it. Okay, so let's say we have a starting structure, let's say closed channel that I mentioned before, and we want to go to this open channel structure, which we don't know experimentally, right? And then, so how do you go to this from here to here? This is we know from experiments, let's say. And I'm representing this mountain and this valley and the lake as a mountain means high energy and valley means low energy because proteins is going under different conformational landscape and has different corresponding energies associated with it. Okay, so the way we do it is called adaptive. We said the method is called the FAST. It's a goal-oriented sampling method called fluctuation amplification of specific traits. And it works with these two different, again, I'm not gonna go through this in the workshop, but just for the knowledge purpose, I think I'm gonna explain it. So we work with this function, it's called exploitation and exploration. So what does it mean is that we want to launch simulations. We're launching a lot of different simulations parallelly, not too long, because if we launch just once straight simulations, then you might go and then maybe get stuck somewhere here, which you don't care about. Or maybe you go somewhere here, we don't care about if you just rely on one single very long simulation. So the idea is that you run parallel simulations, and then you don't run too long, and then you gather the data, and then you ask the questions, is any of my snapshot in the simulations giving me the answers that I care about? And then you gather those snapshots in the simulations and relaunch simulation. And keep doing the same thing until you hit your, get your answer. So this is a way of not forcing it. You're not putting any explicit force, but only the force comes from the selection of, or the bias comes from the selection of the snapshots. and it's called adaptive sampling. This is one of the enhanced sampling methods.
So how are you able these biases? It's not like unbalanced.
No.
Totally
Yeah. So what you do is that you need to have some knowledge of the system. Let's say, for example, opening of a channel, you have designed some distances, pairwise distances, and you ask, this is done in an automatic way. So the algorithm look for the maximizing the distances. Whenever you see some snapshots, it's maximizing the distances, then it select those and then read on simulation from there. So it could be, let's say, for other system, let's say, I don't know, a kinase, a loop opening, then you can design the distances based on that, based on your knowledge.
Yeah, it's also requiring the quality variables.
Exactly, it requires the quality variable, exactly, yeah. But you don't need very precise definition because you're not forcing the system, you're not putting explicit force. So you're just relying on selecting the snapshots which gives you the answer. So it could be a bit more vague. You don't need just one distance, it could be like 100 distances.
It is not like metadynamics and not like umbrella something.
It's yeah. It's more of a no biasing force approach. Yeah, because we have to. So one of the ideas is that we want to preserve the kinetics here, not just the thermal. Because if you put forces, then you lose the kinetic information, and you cannot regain the kinetics once you lose the kinetic information. And then with this approach, you don't lose the kinetic, but it's more expensive to run these simulations than umbrella sampling or metadynamics. Yeah. Okay, well, now that you have all this data, right? So this becomes quite complicated by itself because it could be thousands of trajectories. And this is where this method called Markov state modeling can help to analyze on top of the simulation data. So this is more of an analysis part where you have all this simulation trajectory. And then you first do is clustering of different trajectory, of all these trajectories. And then you just simply count the number of transitions between the clusters. For example, the two different colors mean how many transitions happens from this green to blue, for example. Then you count and then you build this matrix. And it's once you solve this eigenvalue problem of this matrix, then you get both thermodynamics and kinetic informations on this. So this is a very nice review, actually, if you're interested. You can scan this QR code, extremely good review if you want to read about it. And it's very easy to, not super complicated to understand as well. Okay, so why do we do all these things, right? So okay, I can skip this one. What we care about is that we care about not just, let's say, oh, we are in the open state or we are in a closed state. We don't just want that because we also want to see whether this open state we are sampling, for example, this one, is thermodynamically makes sense or not. Because if it's the energy of this state, it's not very stable. If it's like in a very high energy from the closed state, that it wouldn't make much sense because it's just very transient and fluctuating around. So that's why you need to do all this analysis to see what are the meaningful structures you're getting from simulations. And this is what I'm showing here. So this is the one which already we know from experiments, and this is the one we discovered from simulations. And it also has this wider open pore as well. Okay, so now the question becomes, okay, now we found this. It's nice and cool, but how do we know that this is correct or not? Because simulations is, as you said, there is force field could be wrong. Many things could be wrong in modeling, right? So the next question, yeah, this is the thing. Yeah, this was the more of a review paper if you want to read about enhanced sampling. But I'm going to go to this next question now, which is about verifying simulations. So what do you think how we can or we should verify simulations? Again, I'm going to go to this menti.com. I'm going to skip the next question. Yeah, so this one is, yeah. Is there anyone doing experiments here, like wet lab experiments? You are.
In my case, I have very large NMR and crystal structure data source, so I can compare with my metadynamics results with these experimental results, and also based on the energy.
Okay, that's good. Yeah. So you do both then, experiments and simulation. Wow. And you're a PhD or you're a PhD student?
I began.
You began. Wow. Okay, you're quite advanced PhD.
No, I got the messages from the PhD.
Ah, I see. Okay. Okay. Still, yeah.
Not a fancy thing.
What else? Other experiments people do here?
Vitro?
Yeah. Anything? Yeah.
I'm already conducting a 101 experiment in silico with GROMACS simulation. Just in silico.
In silico. Okay.
We will do some crystal structure docking results, proving it with MD simulations and doing it their RMS, RMSF, MMGBS, MMTBS analysis and cross correlation analysis. And today I come for the improve my GROMACS skills.
Okay. Yeah.
Learning background.
Yeah. Great. Yeah. Okay. Let me see from here. RMSD. Okay. Yes and no. Yes, RMSD is the first thing you should check, I think, because to see that things are not blowing up, right? But it's not per se to validate. It's not if it's stable doesn't mean that you did something. Yeah, you didn't do anything wrong, for example, let's say, but it doesn't mean that you have something useful information from the simulation. That's yeah, but that's the first thing you should check for sure. Yeah. Experimental data, yes. What kind of experimental data you think would be useful for verifying?
Estimates.
Sorry?
Estimates of the in vivo assays.
Yeah. Yeah.
Which is about the interactions between the molecule and the target.
Yes.
And between the simulations, we have to very see the time. So based on this.
Yeah. 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 Anyone else?
Binding energy.
Yeah. You can measure binding energy from experience as well. Exactly. Yeah. Yeah. Let's see. Convergence. Yeah, that's a good point. Yeah. What anyone wants to comment on convergence? Convergence. Someone says it's a small one, but it's a very important one. So we can talk about quickly as well. Convergence, yeah. Like something is getting stable. RMSD is one thing. You want to see RMSD getting flattening out, that's convergence. Sorry. Energy, yeah, exactly. Energy of the simulation system, sure, but that would be normally fine. I mean, nowadays things are very stable. That won't be a big problem. But convergence in terms of free energy could be as well. The one I just showed, right? The free energy profile. So for example, this one, I didn't show you the convergence of this one. For example, if I run these simulations I just showed before for another, I don't know, 10 microseconds, is it going to be same or different? If it's same, that's a good thing. Otherwise, if it's different, then how do you even know when to stop our simulation? So that's a really important thing. Otherwise, we might be completely misleading ourselves, seeing that, okay, we're seeing something useful, but at the same time, then I run the simulation next week and then see something else. So that's not a very good thing as well, right? So the convergence is extremely important to test. And there are different ways to check convergence for free energy, which I think, yeah, we won't go today. Yes, but let me also give my example of how I verified my simulations. The first thing is that depends on the system, what can be measured of the system. For example, I was working on this channel. So by definition, channel will have current. So in experiments, people can measure the currents, right? Using electrophysiology, that's the one of the very, or the only, I think. typical experiments people do. And as I capture the open state, I calculate the conductance in simulation as well, because in simulation you can put electric field. You can literally put the voltage across the membrane. You can count how many sodiums, how many chlorides are going through, and then you can use this just a standard formula to calculate the conductance. And you can compare this with experiments. As you can see, that it's matching quite well with the experimental value. That's one of the very first thing I did in my study. Another thing which we care about is the kinetics, right? Because there is open and close, how fast or how slow they open and close. There's also, we know by experiments, we know this experimental value as well. We can also calculate this from simulations as well, because using this analysis, which I showed before MSM Markov-State modeling, you get these kinetic values. And it's not exactly same for sure, but even having this, the order of magnitude similar in kinetic value is quite a good agreement with simulation, given the fact that there are so much approximations goes into simulation. And another thing you mentioned about the assays, right? Which is one assay could be mutations. You do mutations, now you know what is this value here, and then I see, oh, these are the interactions I see in the open states versus the closed states. For example, the details doesn't matter. And we wanna see the mutations. For example, these mutations we did where we muted these residues around it, and then it decreases the kinetic constant here. And also, we also know from experiment as well that it's supposed to decrease. This is the experimental value here. So that's another way to check. We do mutations, which maybe we know beforehand in experiments, or we predict these mutations from simulations, do this in simulation and see in experiments whether it can be verified or not. That's another way to do verification in simulation. So these are all very important things to think about while we do simulations, because otherwise, I mean, we can learn doing simulations like just today, just run some commands and then we can do simulations. But that's not that's the first step, of course, but that's not the useful thing about simulation. Useful thing about simulation is that understanding new biology and how to verify this with cross-validation for different experiments or different analysis or whatever you want to do. So that's extremely important when we do simulations. Okay, yeah, here as well, I found a new opening of a drug pocket. That's another extremely important informations because ultimately what we care about is can we somehow tweak this open state or not, or any other state in a protein? Can we make it design a drug? Can we stabilize this or destabilize this open state using a drug or not? So that's another hypothesis which we built from the simulations, which we haven't tested this hypothesis yet in experiments, but this is something we proposed in the paper because we didn't have, there are too many things to do in the paper. Okay, so now that's my details. Like if you want to contact me later on, but I think I was perfectly on time, even though I didn't plan at all the timing. Yeah, so I guess we're gonna take a break now, right? Yeah. Yeah, sure, yeah, we can, yeah. We can have questions and then we can take a break and then we're gonna go through the workshop together. Yeah.
In the hands-on question, can you tell the details of how we change the MDP parameters? How we can determine these parameters by how many there are so many parameters on MDP files.
Yeah.
How can we choose the right
parameter? Yeah, that's good. I can go through this while I'm explaining. I didn't plan this, but I can go through this. Yeah. Yeah.
You know that your own information is which is how can we define the initial structures each or how long distributions should be
the one I explained, like the mountain and stuff. Yeah. So it's kind of like there is no right or wrong answer, but the standard thing is that we run for around 50 to 100 nanoseconds for simulations and then 20 to 25 replicates. That was normally people do in the field in that particular sort of simulations.
be Bayesian structure.
Yeah. Not all endpoints, but the one the algorithm picks to be the good one. Yeah, to be good one to reach our answer. Yeah. But if you want, I can show you more details because there is a whole code and everything, do everything automatically. So if what happens is that there is a Python wrapper, the Python script, which wraps around GROMACS, meaning that it will launch all the GROMACS simulations on the fly for you. You don't have to do it manually by yourself. Yeah. But this is also in the paper as well. So if you go to this paper it explains how I did everything. So you can also read it in this here as well. Anything from the Zoom maybe? No, okay.
And enjoy your hands-on session.
Okay, so, oh yeah. I think it's working, green, right? Can you hear it? Hello? Yeah, can you hear it?
Yes.
Yeah. Okay, so I have prepared a tutorial, which is one of a basic one. So we're gonna like see how a helix we can form in simulations, and also how mutations in the protein can alter the effects of how the helix is formed. And also I have prepared also the output for this one, but we will first try to do the simulations, and then if it doesn't work at all, then we're gonna go to my output, and then we can analyze this as well. So this is gonna be a kind of a basic one, and so if anyone is wants to not do, it feels like this is too easy for them and wants to do something else, they're completely fine with me. So and I can also help you on that as well. For example, there are more tutorials, like for example this one, which is why this is not I think this is not mine. This thing, I think, is somewhere. Is it the touchscreen thing? Oh yeah, it's here, yeah. Yeah, so this is one of the standard tutorials you can go through if you want to do, I don't know, protein umbrella sampling, protein ligand complex. I think protein ligand complex you're going to go through tomorrow as well. So we don't need to do this one today maybe, but maybe umbrella sampling if you want to explore. If you want to build systems in membrane, normally we use Changui. That's another thing if you want to try. You want to try metadynamics, you want to try, I don't know, umbrella sampling, whatever. So I can help you. So you can ask me specific questions about your research as well. If you are struggling with something, I can help you. Or I'll try to help you, at least. But for the sake of, in general public, so we're gonna go through this tutorial. So I have already posted this one, so you can, so this is gonna be more of a me doing than you're gonna do it, and then I'm gonna help you. So you have to read it through and then try it out. So once you click here, you're gonna see, yeah, I don't need all this. Let me actually quickly, because it's a live link, somehow I put twice the same thing. Need this. Okay, so once you click here, this link will pop out, and then you can just download this one in your computer. And then once you download, then these three things will come out. And then this is the one which is practical. This is taken from one of the class I was teaching last semester in KTH. So it says something about computer labs, this coursework and everything, but it doesn't matter. You can just ignore some of this, like these numbers and stuff. So it explains first some of the basic things like force field and stuff. If you know about it, you can just skip it. And then so we work on this particular protein with just 16 alanine. Alanine is, I think all of you have biochemistry background, right? Right? So you all know proteins in general, I think. Yeah, okay, great. Yeah, if you don't know, it's fine as well. We can also, I can also try to explain some basic things. But so you're gonna open this in VMD. Have you used VMD before? Everyone? Okay, yeah. Okay, perfect. Yeah. Oh, then we're gonna be pretty easy. Okay, all right. So you have VMD in your laptop as well? Okay. Yes, yeah. So we will need VMD because, I mean, we can just do just command line in simulations, but then you won't really understand what's going on in the system. So I think it's very important that we look at things in VMD. So it goes along, GROMACS and VMD goes quite along one-to-one. So we're gonna open this in VMD, you're gonna see this kind of linear chain thing, and then we're gonna use it as the initial input, and we're gonna prepare the GROMACS files for the simulations. Then the simulations are supposed to run for 500 nanoseconds. But I think within this time, we don't have the time to simulate for 500 nanoseconds. So that's why I prepared the files already simulated. But I would like you to try the simulations, running the simulations if you never simulated anything before. So I think it's going to be a really nice experience to try it out. And then there are other that explain something about how the files looks like in GROMACS. Because once you start running commands, you're going to see different files popping up in the system. And it explains what these files means. And you can, again, you can ask me more detailed questions if you have anything about this. I mean, you can ignore the details for now, but it is good to know. But I mean, of course, you won't be able to understand everything, I think, in just 2 hours. And then it goes along different analysis. For example, calculating RMSD, how we calculate RMSD using GROMACS, for example. And then later on, we have also 2 different mutations where you will see once you read through it, you're going to see 2 different mutations. We can just do 1 mutation. And then there are questions like, it affects the simulations, those mutations. And there are questions to understand like why it affects those the simulation the way it is. So I think it's really a way to understand like what can simulations teach us, what we can understand about the protein and thermodynamics and how it behaves. So yeah, so you can just open this file and try to follow this one by one. And because some people might not have GROMACS, some people might not have VMD and everything. I think it's good if you kind of work in a group. It's gonna be much easier than just working by yourself. Not everyone doesn't have to do everything. So maybe one person take a lead and then kind of everyone follows and try to help each other. It's gonna be much easier that way. Okay, and the first thing you need is the VMD in your system, in your laptop. I think we can open. I don't think so. We can open VMD in the supercomputer. I think it's gonna be even slow. I think let's just install it. So you can go to this website. Sorry. Yeah, you can use maybe someone else's. Maybe we're gonna work on together. Also, there is also a guideline for installing the softwares as well here. You can skip the GROMACS part because GROMACS is already installed in the supercomputer. So you can skip the GROMACS installation, but VMD you need in your computer.
Install in on demand.
Why this is not working? Yeah, so VMD can be found here in this one. You can go to this website. You can see, I think, everyone. And then software VMD download. From here, you can go to the latest one, for example, or this one as well. Based on what systems you have, you can just Mac or Linux or whatever, Windows. You can just. So what I would do is that not everyone has to install this. Whoever has surrounding you, just one person is good enough. Just, I think, form groups if not everyone has it. And you can get the VMD from here. I can also put this link in here as well. In Mac, installing VMD could be sometimes trouble because they ask for this permission. So I can help you if you're struggling with Mac. Sorry. Yeah, sure, yeah. Let's just get VMD first. I think that's the. Yeah. I can wait for 5-10 minutes, I think. Yeah. I tested that. Yeah, VMD. Yeah. So I'm gonna wait for 5 minutes and then ask if everyone has it, and then if not, we can help. You can ask me, and then Sudarshan as well. He's also can help you. So both of us can help you in your hands problem. Yeah, if you have it, yeah, just go to the practical. You know where to find it, right? In Slack. Yeah. And if here. Unzip, yeah. Just unzip that one. Yeah. Exactly, yeah. Yeah.
Tamam. Tamam. Şimdi yardım. Tamam, çok teşekkür ederim.
Oh, the accounting, right? Yeah. So is VMD is kind of installed mostly? should we move ahead with the next one? You can raise your hand if anyone needs still help. Sorry? Yeah, no, I can come and help you. Yeah. Yes, yeah. Ah, okay. Going through this thing. Yeah, all right. Yeah, exactly. Yeah. Yeah, I think it's fine for me. Yeah. 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 Yeah, I'm gonna show this next. Yeah, I think it is. Yeah, no, I'm gonna show this next. Yeah. Oh. Yeah. Hopefully I didn't break anything, right? Okay. All right. Okay. So next is that we go through the file, right? So we're gonna follow this practical as you might have already downloaded it from the OneDrive thing. Everyone has access to this PDF, right? Okay, perfect. And then, so you're gonna read through this yourself. I'm not gonna go through it myself. But what I'm gonna show is, so you might see somewhere that of course the VMD you can open it yourself because you already have downloaded the file. So if you unzip this one, and then you're gonna see here ELA16. So this is the file I'm talking about here. This is the one, ELA16 PDB. So this is the one you can open and see in VMD. But then once you have to, for simulations, we're not gonna use VMD just for visualizing your molecule. For simulation, we're gonna use GROMACS. So you might see the ZMX, let's say one of the GROMACS command. And for this one, you are going to do this in your supercomputer, which is the cluster you have. I think everyone has access, right? Yes. Yeah. Okay, so I'm going to also access myself. So because now the files you have downloaded is in your local directory and the PDB files, the simulation you're going to do in your supercomputer, right? So you need to transfer them whenever you just need to transfer one thing there, that's it. Let me show you how we're going to do that. Wait, what is my... Yeah. I want to... Okay, so this is the interface of the supercomputer. So everyone can log into the account, right? Anyone is struggling? Yeah, so you just type this. I don't know how it was. That was mine. Everyone has the same instruction for logging in, right? Or everyone has the same instruction for logging in, or okay, yeah. So okay, if anyone cannot log in, just raise your hand. All right, so here I have files here, but you might not have anything here. No, not all. You don't have to transfer everything. No, you just transfer this thing, ELA16 PDB. So that way you can transfer in your local desktop. You can just do SCP-R, which means copying your files. This particular PDB. This is your account name. This is mine. You can be different. And this is my home address. This also could be different for you as well. Just use this command and modify it accordingly and transfer the file. Just one file. That's it.
from local to my scratch, is it also file?
You have a scratch space as well?
Zip it practical file from local to my
Yeah, that's fine. Yeah, the whole thing. Yeah. Yeah, you can also take the whole thing, but you don't need the whole thing really. Okay, so one thing before you start doing, just one thing, you have to replace this GMX with GMX/MPI because that's how it's set up in your cluster. You have to do this thing. Oh, wait. No, you have to do also module as well, actually. You have to do, to get GROMACS in your supercomputer, you have to do this. Module load this, which means that you don't have to install it yourself because the admin has already installed there. Module load this. I can also post this in Zulip as well. I'm posting everything in Zulip so that you can directly copy paste. And then after that, to and when you go through this tutorial, you replace GMX to GMX_MPI. That's all. Yeah. Otherwise, it won't run.
Can you show again from starting how to extract files?
Yeah. So did you see this one? So I posted this like last night, I think. Yeah, yesterday. This GROMACS tutorial, formation of a helix. You just click here, and this is where you get your thing.
And then how do they take this from here to the supercomputer?
Yeah, once you have this one in your computer, you can just go to that particular directory wherever you have it in your command line, and then you just do scp -r this command. So this is gonna copy the PDF file to the supercomputer. Okay.
Maybe you can just expand that particular command.
Yeah. So this is the command. Let me put it maybe in the middle as well. So this is the command. So you might have to change the username if you have different username. I don't know. So based on what username you have. Hi. So is this everyone is running this in their home directory? Okay. So not here, not ARF home. Okay, everyone has their own username. Okay, yeah, okay. You still have. Okay, you're gonna do different one, yeah? Sure. Okay, yeah. Okay. You have to download GROMACS. GROMACS is you have to just access the supercomputer. So in your where is the terminal? No, the terminal. Yeah. You put your yeah. Oh wait, no. We have putty? Ah. Yeah. Can you go to the oh you have to also use this thing as a VPN.
Yeah, you can. I don't know.
This one?
No.
Yes. Download. Yeah. Oh, that's a VMD, yeah, sorry. You already have it. Yeah. So can you open this one? So you should have a file here. Okay, I can send you. Sorry? Okay, let's make one thing. I just posted in give me one second. I just posted in this one. In Slack. You have Slack? You don't have Slack? Oh. Yeah. I don't know how else I can give it. No, you will need Slack to access these things. Otherwise, you won't be able to open this thing. Maybe you can pair up with them or something. It's gonna be easier. Yeah, yeah. Okay. Yeah. Sorry. Do you need what? Yeah, you need the supercomputer account. Oh, you want to run it in your computer? Yeah, you can run it here as well if you want to install. Then you have to install it yourself. Yeah, no, yeah, you can run this here as well. Yeah. But you have to install the GROMACS thing yourself then, yeah. I think having in the supercomputer, they have installed everything for you, so you don't have to install GROMACS yourself. Yeah. No, it's fine. It's fine. No, it's fine. It's fine. It's fine. Which one? Did you downloaded the files? Yes. Where are the files? No. Yeah. No. So you need the. So have you downloaded this one somewhere? So you have now here downloads. So here you can go and then new molecules, browse, downloads. Sorry? No, I'm from Assam. Oh. You're from Odisha as well? No. Yeah? I'm from UP, okay. And you were here working? Yes. Okay. 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 And you're doing simulations or?
Actually, I'm interested for drug interaction.
Okay.
Some of my friends do this type of work.
Okay.
So that's why I can do it to know about so that's why I can by myself.
Yeah. So this one, Ella. You can select here, then you can load. Yeah, see? Now you can do next one, yeah. Okay, one thing, if you have gone through GMX, the first command is that once you write this, it might ask for this charm 36FF force field, which doesn't exist because it depends on how we install this. So you just use instead number 6, the amber force field. These are all different kind of force fields, which for now, for this purpose, it doesn't matter. You can literally choose anything, but just choose number 6 for this purpose. And also, if you have questions, which you can also ask in Slack as well. I can also answer directly if you don't wanna. Okay, Zoom. Number 6. Okay, one thing. If anyone is still struggling with installing anything, I would suggest to just move on and pair up with other people, because otherwise we won't be able to finish anything today. Can you hear me? So if anyone is still struggling with installing anything, just move on and pair up with other people. We should move on faster, otherwise we won't be able to finish anything. You can figure this installation 6 later as well. That doesn't matter. Okay, I think for now you should be at least in this phase of the tutorial. Is everyone in this phase of the tutorial? At least you can move on as well. You can be further as well, but at least here. Sorry? Yeah, P3. P3, yeah, exactly, yeah. 1. Yeah. Sorry? Yeah, you choose 1, yeah. We don't have charm 36 in the system right now, so just choose number 6. That's Amber. Doesn't matter. Yeah. 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 But to be honest, for this tutorial's sake, you can choose anything. Doesn't matter. But for your real experiments, it matters. But for now, just choose anything. Yeah. So yeah. So for yeah. Yeah. Latest one, yeah. 6M. Yeah, exactly, yeah.
I use this all the time.
Yeah, no, that's perfect, yeah.
I don't know why or change or important.
So yeah, it's important to choose the latest one. The charm 36M is the most latest one. And then yeah, those parameters are updated, yeah. Yeah. And so for me, if I do simulations with membrane, then I typically choose charm 36M because it's more optimized for membrane kind of simulation with protein and membrane, proteins, membrane, and everything. It can simulate anything. Yeah. 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 M. Yeah. Yeah. Or also you could choose Amber as well. Amber is, in my experience, Amber gives me better results for protein ligand interactions.
calculation.
Yeah.
We usually use average.
Yeah, that's yeah.
For GROMACS, I use the difference.
Okay, yeah.
Everybody is more useful.
Yeah.
I think.
Yeah, because I think they are also for small system, they're faster as well, I think, than GROMACS. Yeah, yeah, yeah.
Okay. Each one. Yes. For example, there's more
Number 6 is one. Number 6, another which doesn't exist right now here is this 19 SB. That's a more recent one. You can have you used Charmgui?
Yeah.
Yeah, Charmgui you can. Yeah, the Charmgui you can get the those. Yeah, yeah. Yeah, yeah, yeah. So yeah, so I also use normally Charmgui, and then yeah, as well. So there, I think the 19 SB, that's the most recent one. So I would use that for Amber. Yeah, yeah. Well, I think the way I'm doing here is normally we don't do it because with membrane you cannot do this way at all. Membrane you have to use Charmgui. Yeah, yeah. Yeah, 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 yeah. Which was a very simple one because just to make it simple. Yeah. Yeah. And what kind of simulations do you do? Yeah. Okay, so you're quite experienced already. Yeah. Yeah. So you might be useful to tomorrow sessions might be more useful for you. Yeah. Yeah.
Same as I don't change anything.
Yeah. Normally MDPs, I think they're updated. MDP is the MDP file that sometimes could be outdated. Some of the terms there. Yeah. 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 Yeah, yeah. Yeah. Actually, I'm gonna go to the MDP. Actually, someone else asked as well. I'm gonna. Yeah. I'm just waiting for people to kind of go through until MDP. Yeah. Okay, so for now, then I assume that everyone is in this GMX solvate phase. If there is, if someone is not in this stage, you can raise your hand and we can help you. Because you're gonna move on to the next one after this one.
I can visit somewhere. I can share that. If you ask no, he does.
Yes, I'm and I get some problem.
You have modified version of non-natural which you already have, but you are not sure about
just I tried to
And once you do the solvation, if you want to visualize your system like the way it is, you have to transfer your files from the supercomputer to your to your laptop, right? Because you're running this in a supercomputer. Now you have to transfer to this in your local machine, which is the same thing again. What you do, you go to your terminal. Before you push your files from the laptop to the supercomputer, now you're gonna do it the other way around, which is that let's say this file exists here. So you're gonna do SCP-R. This is your login name. That's your address, and then wherever your file is in your supercomputer. And that way you find the location is that you just do pwd. This is where you get the path for your files wherever you are. And then go here and then just put the file name. So let's say this is my account in the supercomputer. And then I do pwd. This is my path, and this is the files I want to transfer. I don't know, let's say this one. No, I'm just saying, okay, sorry. So you want to transfer this one, solvate.grow. Okay, this is the file you should have in your folder. Okay, and you want to transfer that. To do that, you check where your files are, and then just do this, and then just write this dot at the end, and then you click it, and then you can ask for why. Connection closed. Oh yeah, it's gonna ask for your password and stuff, then it's gonna transfer to your directory. Okay, this is the command here. So I also put this in the Slack as well, this command. You have to change it, your username and stuff here, based on what you have.
Sorry, I can't get all this in my computer.
Oh, that's fine. You have GROMACS in your computer?
Yeah.
Oh, that's fine. That's perfect. Yeah. Doesn't matter. Yeah. No, it's fine. That's for practicing. Yeah. I already gave you the answers, like the output as well. It's in the file. So I'll show it. Yeah. Okay, so I assume that everyone has the solvation until. So now I'm gonna explain a little bit on the MDP files. These are quite standard thing for like regular simulations. It could change based on what you want to do. But so after this, so now that you do the solvation, right? And then you have the system basically you want to simulate. You have the protein, you have the water, you have ions, everything there. So now you're gonna do the simulations where first of all, you do some minimization because sometimes there could be some overlap of atoms. So you're gonna kind of relax the system. When you do this minimization with this thing, and then after that you do simulations with this command here. But for this, you need the most important thing you need, not just the grow files and stuff. You also need this MDP, which means that it's gonna tell you the parameters when you run simulations or minimization. And so you don't need to really change anything, even though you in your own research. It's very like these are all optimized parameters. People have tested it before, so you can just trust them. Some specific cases, if you run some other kind of simulations, then yeah, there could be different scenario. But in general, you don't need to worry about it. And like how many steps of minimations, this is 5,000. There is no, you can run longer, but nothing will happen. So that's kind of a standard thing, just run for 5,000 or maybe 10,000. It won't change much. This is not something, oh, if I run for 10,000, is the big mistake or something? No, you can run 5,000, 10,000, doesn't matter. But you don't want to change these values though. These values has to be what it is because these are tested. And you could actually read in GROMACS. So if you go to this, you can write in Google called GROMACS MDP options. So every single things in the MDP is explained very clearly. So you can, there's a huge lot of big document here. You don't have to read everything, right? So for example, I want to see, I don't know, n steps. You just copy this thing and then paste here and see. Yeah, here is explained what it means. -1 means no maximum. Yeah, 0 maximum number of steps. Yeah, exactly. How many steps you want to do the minimation for maximum steps? And then let's say you want to see, I don't know, how many times, let's say, frequency of saving your file. This is the frequency. This would be in. So what do you think? This is 1,000 means what do you think? What is the how frequently are we saving our simulations? You think?
Taking a snapshot.
Yeah. So every 1,000 time, right? Yeah. What does it mean? Like 1,000 means how much in the time you think? The real time. What is the time here?
Picoseconds, I guess.
Yeah, we simulate in 0.2 picoseconds. So you just multiply that, and then that's the number you get, right? Sorry, femtosecond. Yeah, yeah.
Sorry.
And then what else? Yeah, so you could check this. This is very. Yeah.
I wanted to ask, like normally in default mode, GROMACS uses the steepest descent for the energy minimization. But
yeah.
Many people use other techniques such as I forget the name, but there are other minimization techniques.
We can actually check here as well. Let's see.
How can I check what it's using and how can I change it?
You can just so you can what you can do.
So maybe like I want to do 5,000. 5,000. Yes. I forget the name. Sorry.
Okay, so the way you can do, you can just search this. You see integrator, right? This is the one it's defined. So you can search the word integrator here, and then you will see. Oh my God, there are so many. Okay, here, yeah. So this is the one options. You have different options here, which shows the. So you can choose from here basically what you want to. We are choosing right now here, but I mean, yeah, steep addition is the most typical one. And I always use steepest descent. I never use.
Now I remember conjugate gradient. Like maybe first of all using the steepest descent and turning it into the conjugate gradient so that we will have more biologically relevant data structure.
Yeah.
So I will do that in the leapfrog integrator.
No, yeah, exactly. Yeah. So you first, let's say, do steepest descent, and then you have your output grow file, right? And use that grow file to do your other thing, whatever you want to do. Yeah. You cannot do both within a same MDB file. I don't think so. You have to do it step by step.
Okay.
Yeah. Yeah, minimization is normally same. Yeah. Yeah. And also, when you do membrane, normally you use Changui, right? There you're gonna get everything, all the MDP. Yeah, yeah, they will give you all the things there, and then you don't need to change anything. You just use. No, you can use your own MDP as well, but those MDPs are recent and optimized, so you don't want to change them. Just. Yeah, yeah, because in your PDB file it will be written your box size or grow file, so it will recognize from there. Yeah, yeah.
I don't need to change anything.
No, in Changui, no. No, Changui is extremely optimized. Yeah, yeah. Okay, so yeah, so that's for minimization. The same thing also goes for running the actual simulation as well. For simulation, you would need, let's say, the pressure coupling, temperature coupling, pressure coupling. Yeah, these are all very well optimized parameters. You don't change anything. You just use you don't want to change these values or anything. I mean, for example, reference pressure is 1, right? You don't want to no one simulate this in 2 or something because 1 is the most natural how it exists in our in the nature. So this is
On my I couldn't use another number.
yeah.
I used POC.
Video scale, yeah.
And I was changing it.
Yeah. No.
All right.
No. And if you have if you have membrane, you might use semi-isotropic, not isotropic, right? Because in the membrane normally the pressure should be different along the. That's fine. That isotropic is fine. Yeah. And yeah, so my point is that these MDB files are not meant to be tweaked too much. Only thing you tweak is like how many, what is the step size, how frequently I want to save, because you don't want to save too frequently because it's too much data. Normally I save like 100 picosecond. Sometimes even 1 nanosecond. If I run like microseconds range simulation, the 100 picoseconds. If I run, let's say, too much, even like tens of microseconds, then I run save like 1 nanoseconds. But yeah, that's kind of the balance you have to kind of choose. And also you have to be careful if you save these simulations in that frequency, that simulation in other frequency. You have to be very careful about when you analyze things. Otherwise, you're gonna mess up the time scale completely. So that's gonna be a big mistake. So I think I would say this is the very important parameter to keep track of, like one of the parameter. And I mean, if you do other simulations, like I don't know, their meta-dynamics or like whatever other kind of enhanced sampling, then there could be things coming up here below things, but the above things are always like constant, similar. And normally, I think if you are more of a user, normally the practice is that we use CharmGUI. That's kind of the server. It's much easy to build systems, right? And they give you the MDP files and everything is very well optimized. So you can just very like recent and everything. So you can just trust them and then just run.
I have 2 questions. First of all, in equilibration parts, there are some distinct changes to MDP file to NVT and NPT.
NVT, yeah.
There are distinct differences, or we can use the same.
So normally, no, we want to run in NPT, not because we don't want to keep the volume constant. NPT will be where we fix the volume. That you do normally when you equilibrate the system. You're gonna relax the system a little bit because you don't want to have too much fluctuation in the box. Initially, you might do NVT at the sums, but when you run production, you always do NPT. Yeah, you never do NPT unless some weird restraints you put or something like this. Normally, you should always use NPT.
I use always NVT and NPT same time, same MD. And
but you cannot use both at the same time. Either use NVT or NPT. Because like the way you use NVT would be if you don't have this pressure coupling. If you don't have this pressure coupling, if you remove this one, that's NVT. If you turn it off.
By deleting this part, it's an NPT.
Yeah. Yeah.
Oh, okay. And also, I've never used CharmGUI before. I prepare complex MD input files by using CharmGUI.
CharmGUI, yeah. Have you used it before or you haven't?
I have.
Yeah, I would suggest you to do CharmGUI, unless you wanna build like hundreds of systems at the same time.
I think just for CharmGUI, for membrane protein, membrane.
No, you can use for solvent as well. Yeah, it could be for anything. Why I suggest this? Because like this is like if you go to input generator, this is not part of the tutorial, but I mean, I'm just showing you the way normally it's done. Let's see. Anyway, don't want to. Yeah, I don't know, it's not coming. But yeah, no, in the input generator, you have solvent as well. Yeah. It even builds a weird shape. Yeah, they can do a lot of different things.
In solvent part, we can integrate complex files, complex files by using chunk.
Yeah. Yeah. You can do with chunk everything. Protein ligand, just protein membrane, anything. Just membrane. Yeah. Yeah. Okay, so now we're gonna let's aim for this part here. Running the MD simulation. You don't have to run for long. I just want to make sure that you know like how to that everything's working basically. Even though it doesn't work, it's fine. We're gonna move on, but let's see that we can reach until here. And then after that, there is the analysis part for the last how many we have until 5:30, right? 1 hour. Okay, yeah, we have enough time. Yeah. GMS Okay, so sorry. So when you want to run this one, I would suggest that you don't run in your login node because right now in your supercomputer you are in a login node, so you don't want to run simulations in your login node because it's gonna be the admin is gonna be pretty angry because you don't want to overpower that. So what you're gonna do is that so because we are working with everyone is used to like a slump system or running in the queue? Maybe not. So what we do is that so because that's machine, it is shared machine. So it means that like thousands of people are gonna share at the same time, and they have a lot of different machines, like I don't know, thousands of nodes or something in the system. And you're gonna run in some one random machines there, but you cannot just run just typing this command because if you just type this command, it's gonna run in the thing you are only in, which is the login node, which you don't want to run. So you want to have this file, and the way I got this file is from I got it from somewhere in the system. It's supposed to be, let's see. Okay, so if you search in Google Truba GROMACS, there is a very nice tutorial for GROMACS in Truba itself. All the thing I said, like all these GROMACS where it exists, everything is here. So if you go to this here, so that's always a good idea. Normally GROMACS documents are always in the supercomputer because it's very highly used in general. So you always find some information about how to run GROMACS in any supercomputer almost. So as you can see, the GROMACS is here. This is the one for everyone basically. Here, if you do ls means I just want to view what the files here are. You can see. Wait, I thought it was here. Wait. No, maybe it's here. I think I did module load. Oh, I didn't do module load yet. Okay.
Yeah.
It's here, right? I think I'm gonna find here. Let's see.
It's under scratches.
Yeah. Yeah, exactly. I did this one. Yeah, it's here. If you do this, you'll see. Wait, I think yeah, you'll see that these are the files here. This is the one you're gonna see, which already provided by the admin in the system. You can just copy paste this file. You can just do this, CP this file in your folder. This won't work in your computer though, because in your laptop is not a Slurm system. It's just your own laptop. If you are in your computer, then you have to just run this command like GMX, this thing. But if you are in the supercomputer, just copy paste this file in your folder, which I'm gonna put again. So just copy paste this because this is the one which can help you to run the simulation. So open this file. Once you copy, I'm gonna quickly go through what it means. Sorry, yeah. You don't have this thing here, MDP file. Oh yeah, so yeah, sorry, actually I forgot to explain. So you also have to copy the MDP file as well, because MDP files are not there. So the same way you copied your PDF file, copy the MDP file as well. So here, as you can see, we copied this one, right? The same thing here as well. So you do mini by MDP, and then minim MDP, and then MD MDP. Just copy these two files to your supercomputer. SCP this.
Where is the MDP file?
Yeah, MDP file is in your computer. So you have to copy this to your, with this command, SCP this, and then copy this to your supercomputer. That makes sense?
Dosya şey indir. Bilgiyi indir. Ben başka bir şey
So this is exactly, yeah. So this is here. Once you extract the file, initially you had this MDB, this file as well, along with this one. Yeah.
Yok hocam bak bir tane mail var. Bütün bilgiler oluşuyor.
Tamam, o sevgili istedim lütfen yanına.
Orada kontrol etti arama yapın.
Yaptım.
Bu şeyde.
Özellikle yapmış.
Turba HPC sistemle başlamıyor. Turba HPC sistemle başlamıyor. Temporary account takes for exploring from.
Ben size atayım. E-mailiniz ne?
En sol ortalar.
Efendim?
En sol bu. En sol. Atalar. Ve. Atalar. 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 Ve. Ve. Şunların dersi. Pratikler.
En sol atalar ve.
Sistem bir sistem. Ben unutmamış.
Peki Excel'de hangi Excel'de hangi ama ben bir şey
Okay, so has everyone run their first simulation? Anyone still needs help running simulation? Whoever has finished, they can continue with the below the follow the tutorial. But what I would like to mostly focus on in this last part, I mean, everything here is fine. It's animated and everything, you can try it out. But most important thing is this last part where we start with in the last task of this lab, we will monitor collective variable, a degree of freedom. So this is the part which is gonna be most interesting one. But for that, you need this Python package to analyze because we wrote the code to basically extract the information. And but for that, you don't need to run all these simulations because it needs quite a bit of long simulations. So I already have given you the output files. This is called long trajectory. There's a folder called long trajectory where you can find, for example, let's say ala.gro, that's your input, the initial file. And then if you go to ala.xtc, that's your trajectory, the simulation trajectory. So this is the file you're gonna work with. And you can also open this in VMD as well. Just try to open both in VMD and then see how it looks like first. And then also try to run this Python script. But for this, you would need to have a Conda environment or even not, but at least you need the Python to have this so that you can call this few packages so that it can do the analysis for you. You can either do this in the supercomputer itself or in your laptop, whatever you feel. If you are doing it in the supercomputer, then you have to transfer these files to the supercomputer and then do the analysis there, and then transfer it back, the outputs of the analysis, to your local computer. But I would suggest, if you could, I would suggest you to do this locally in your laptop because it's gonna be easier. And in the tutorial itself, it tells you how to do the conda and everything to install. And then at the end, there are a few questions which asked in the thing. So this is the most interesting part where we're gonna go through to understand, because for now you just run the simulations. So it's important to know also what we can see, what biophysical understanding we can learn from doing simulation. This is the most important part. So no, so you're gonna run this one. So in your so what you can do, you can just copy this thing. You can also do both as well. You can prepare. This is the file which prepare the system, right? So this one you can also put it here. Just do, let's say, GMX. So just you have to just replace this thing. So this one, and then again same thing. Conf.md. I just replace this whole thing. Yeah, like this one. You can run both at the same time. You can keep everything else same, doesn't matter. So this is how your SLAM file should look like. No, don't wait. If you are able to run the simulations, just stop it, forget about it, and use the long trajectory folder. Yeah, there is a folder called long trajectory. So here, if you go, the file I showed before, it very initially, you have this long trajectory folder. And yeah, so I would suggest to do the analysis locally in your laptop because it needs Python. It's gonna be easier. Also, it gives some plot as well. So it's gonna be easier to visualize the plots. Yeah, yeah.
Same thing
Mm-hmm.
as yours, but I can.
What does it say?
Account.
Oh, account. Yeah, that's right. Yeah. Yeah, can you go there again? Open it. Yeah, yeah. So you just delete this thing.
Which one?
Yeah, this one.
This one?
Yeah. Yeah, that's it. I think it should run. Yeah. Let's do SQUU. SQU. SQ. Yeah, it's running. Yeah. So it might not, you might have a problem with the Slurm script about the account. Minus A, just delete the line, basically.
Yes, it's wrong to execute. I just
What is the problem? Oh yeah, because this needs the GROMACS to be there. Show me the file again. No, GROMACS is fine, I think. Yeah, MD analysis. Have you installed MD analysis? Yeah, I think in the PDF already explains. Just pip install MD analysis should work. Yeah. Okay, NAMD. Okay. Prodi. Okay, I see. Yeah, yeah. I mean, if you can, yeah, I think it's for this one. I think you can just install this quickly. I think. Yeah.
Have to find any details about learning MD and energy calculation. I have a problem in the pre-energy calculation after GROMACS because there's no, like I use this one, there's also an embedded pre-energy calculation program. But after GROMACS, how can I calculate the pre-energy in live way?
So what kind of pre-energy methods are you using?
I use GMX, MMGBSA.
Okay.
But it is some problems, I think.
Yeah, I mean that's not a free energy. That's just like a approximation of energy.
Yes.
Yeah.
But you use rods of the GROMACS and the
Yeah.
free energy calculation.
Yeah, but the free energy, I use PyEMA. Yeah. For Markov set model. Sorry? No. No, not that's PMX. That's for calculating free energy of protein and ligand. So it depends on that's what you want to do?
Yes.
Okay, that would be PMX, yeah.
Like for GROMACS.
Yeah, I would say so. Yeah, but it's not just running standard GROMACS though. They do like it's called they do a more sampling of different states, and then it's called non-equilibrium pooling, and then so if it's reliable, I mean, if it's not too complicated system, then I would say it's quite reliable.
I actually not want to know not complex. Just simple.
Just protein.
Yeah, protein and ligand.
Yeah, protein and ligand. Yeah.
It's good for.
Yeah, I think so. Yeah.
You use for membrane?
Yeah, you can use for membrane as well. Yeah.
All right.
You can actually ask him more to him actually. Yeah. He's gonna. Yeah. 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 So. No, it's fine. It's fine. We can talk. So temperature. Sorry.
Maybe briefly.
Yeah. So, I mean, temperature is just room temperature, right? So 27 degrees Celsius is room temperature. So normally people use 300 Kelvin, or sometimes I also use 310 because that's a human body temperature, 37 degrees Celsius. That's human temperature, so 310 Kelvin. I mean, there is no right or wrong answer. Normally people use 300 or 310.
Get some results and usually like to compare.
No, I think it's fine. I don't think it's.
There is no significant difference.
I don't think it's just 10 Kelvin is very less energy. It's very few energy. I don't think so. It's gonna be different. I mean, if you compare one simulation versus another, there might be difference. But if you compare a much more data, then it should be relatively similar. Multiple replicates for both. No, I wouldn't do that. Just stick to either 300 or 310 and just. But I think it's important that you stick to one temperature for your whole like project. You shouldn't choose between different things. That's gonna be a problem. Like so actually I don't understand the question.
Why did we set up these permits for our experiment if that won't be any change?
I mean, it's like sometimes what happens is that, example, some bacteria, thermophilic bacteria, they lives in very high temperature. Like, so it depends, right? Sometimes you want to simulate in higher temperature for special cases, but this is a general case. In general, this 310 is standard. Yeah. And for box size as well, it's normally the rule of thumb is that you want to have at least 10 angstrom of water between the protein and the thing, because there is a cutoff of electrostatic cutoff of 12 angstrom, right? If you use less than that, this protein might see the other image from the periodic boundary. So that's why you want to have at least 10. And you can have more as well, but then you are just increasing the size for no reason. So you don't want to do that.
Ion.
Yeah, it could.
General, we use
50. Yeah. 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 Yeah, because that's what in our body typically is. Yeah. But it also again depends on the specific question you are looking for. If you Exactly. If you really care about your ion concentration, then you want to have specifically, because it's not always 150, because in the membrane, you have different concentration, right? Typically outer versus inner. So if you really care about that, you want to carefully design that. But normally, we don't care much about that in the questions we're looking for. So that's why, yeah. Yeah. Yeah, box size is like, as I said, you want to have at least 10 angstrom of water padding of the protein, and then 10 angstrom here for water. But that's already there in the SAMGUI, so you can just use the standard thing. It's fine. You don't need to copy anything. You don't need to copy anything. Your simulations are running, right? So yeah, everything is in your locally as well. So you just use the local files. Yeah, just forget about this one. You don't need to copy anything. Everything is there. So go here. No, you should have this one. No, you should have this file, no? This file, long trajectory. Sorry? Yeah. Yeah, answers are here, yeah. Okay, so at this point, if you are still struggling with, sorry? Oh yeah, perfect, yeah. 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 Have you also looked at them in VMD as well? Have you looked at them in VMD as well, opening? Yeah, no. What I'm asking is that, so now I think if you look at the tutorial, so now what the questions I think at the end is that, why do you see the difference? So that's the question is we are trying to understand. So you want to look at these plots and your trajectory and then try to understand why these mutations is causing this behavior. Yeah. Yeah. Yeah.
My job is finished, but
no, long trajectory, you have to download it. Like
For our results,
uh-huh.
we don't have we also can see should have our results, right?
Yeah, which means it might have crashed. Let's see. Yeah. No. Yes, L. What is L? Yes, can you close this file? Okay, we did the minimization.
This is, isn't it?
Yeah.
Output optimization.
No, this is the input. You should have a
Out.
no.
I ran the minimization step.
Did it work though? Did you see it's running? So what I would suggest is doing this. Just run. What is it?
I can run.
Yeah. Yeah, so just type this thing complete, yeah, copy paste and then just type here. Just type. Wait. Yeah, then go there again, the PDF. Do this one, copy and paste. Yeah. Oh yeah, so you have to run this in the Slurm system. Just open this file. Yeah. No. This one. GROMACS. So this is not, this you have to, there because your minimation is failing, that's why. So comment on this one. Yeah. Can you put a pound symbol there? This hashtag thing. Yeah, same here.
It should be hashtag.
No. I'm just commenting out for now, and then I'm going to run minimation here. So go back. Take the, yeah, take this one. Go to your PDF file. Yeah, sorry, PDF. Copy paste this one. Yeah. No, not here, because this is the problem also, right? Yeah, exactly. In Slurm. Put it here before exit. Yeah. No. Exactly, and so replace this with zip MPI Zmax thing. No. So you also always want to have this thing here. Copy and then yeah. Now save it. Now run this one. Exactly, yeah. It's now it's waiting. It hasn't run yet. Now it should be doing minimation. Yeah. It should finish quickly though. I think it's gonna finish now. Now you should get the grow file from the minimation. No, it's fine. I think it has to finish first. Exactly, yeah. Yeah, okay, now do L, sorry. How to do this? Yeah. So now you have to, I think so just use this. Okay, give me 1 second. So yeah, exactly. So now comment this one and comment out this thing. So now it should run. What is your wild type one? Let's see. So this is 2, right? There should be one more up, right?
This is the glycine.
Yeah, only glycine. So how? No, so just LR16. How does this one look like?
This one.
Yeah. No. So there are 3, right? One is LR16, LR15 Gly, LR15 Pro.
Yeah, this is the Gly one.
Yeah.
And this is the Pro one.
Yeah. And where is the just LR16? Yeah. So the idea is that LR16 is the wild type, and these 2 are mutant. So you wanna see a difference.
I cannot plot this one because there's no DD file. This file.
Okay.
So I cannot.
But you're already seeing the
Yeah.
That's perfect. Yeah. Yeah, so then you can compare these three and then try to understand. You're gonna see a pretty big difference. Yeah. Okay, yeah, yeah. Then you can just start analyzing from this long trajectory thing. Oh, I mean, this will gonna, I think I just ran for 1 nanosecond this thing. So it's gonna 1 nanosecond, but that's not enough. This one is like 500 nanoseconds, the long trajectory. So you want to analyze there. That's where you're gonna see something interesting. Sorry?
How can I start this?
Yes, of course.
Which one? Long trajectory? Long trajectory is in the thing you just download. So here, practical. This one, practical. Long trajectory here. Everything is here. Yeah, this one.
So now it's in the laptop. Or
Yeah, it's a laptop.
It is a laptop.
Yeah, yeah.
There is not in the PC.
No.
So we can use on laptop.
Yeah, you can use your laptop. Yeah, for now. Yeah. Hi. Where is it here or where is this one? This is in the supercomputer, right?
Yes.
Yeah, that's why.
In Windows, I should do this.
Yeah,
Okay.
Otherwise, you can also, you see this XVZ, you can transfer this to your file as well. If you want to run there, then once you have the output, you just use this output transfer to your locally here so that we can open it.
Okay.
Once you have the plot.
Yes,
yeah. Or you do everything in locally if you have Python locally.
They were using some guy in Ember. They were using CPP trash.
Yeah.
That was
CPP trace. For what purpose?
I believe it was, if I don't remember wrong, taking visualization in terminal possible.
Okay. Yeah, I mean, I could have, yeah, sure. But I think I didn't prepare that one. Yeah, normally also what you could do actually, yeah, I mean, if you, do you have VS Code? Do you use Jupyter Notebook, right?
I will use that. Yes.
Yeah. So you can also connect through Jupyter Notebook to the supercomputer itself. And also you can visualize directly there. But I mean, I think it's gonna be a bit complicated for everyone to sync in. That's why I didn't try that. Yeah. And I thought it's gonna be easier if you just transfer or like just do it locally. Yeah.
I did not. Okay, that's why I asked for it.
Yeah, no, those are the options as well. But I think it's the easiest thing is that if you do want to do that, there's a software called VS Code. That's a very universally used software. That way you can just go back and forth between supercomputers very easily. Yeah. It looks something like this. I can so this one. This one here, you can it's called VS Code. You can log into any supercomputer directly and then use Jupyter Notebook there. Like it visualizes in your local computer all the time. So you don't have to transfer back and forth thing. Yeah. Yeah. 15 minutes is okay. 1.5, right? Yeah. Okay, so whether you're able to run the simulations or not, what I would suggest is that just forget about the simulation and then go to this page number. Where's the page number? Okay, the one before the last one, where it says this line right here. In the task of the lab, in the last task of the lab, we'll monitor collecting variables. So this is the one you want to focus on. And then to do that, you have to use this long trajectory folder to analyze this. And there are some questions. If you read through this, just read through this, you're gonna see some questions here. And these are the one which mostly very interesting question in terms of what happens in the protein. So let's try to go through that as well in the last 15 minutes.
Or just run the analysis.
Yeah, just run analyze aids. Yeah, forget about the clustering. I think we don't have time. Yeah, I think so. Yeah. Yeah, you can also email me as well. I mean, later as well if you have any questions. Yeah. Yeah, yeah.
Is there any way to optimize the solvation model?
No,
I don't want to waste my time.
With some way. Yeah, that's the only way, I guess. Yeah. 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 But before membrane, no. It's very complicated. Yeah, membrane is complicated. Yeah. That's why there is Chamgui. Yeah. 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 Well, I mean, there is, if you want to keep the same membrane, yeah, but you have to do a lot of coding and see. But yeah, I think it's better to use the Chamgui. Yeah. Yeah. Yeah, it takes time. Yeah, I mean, it depends. If you, let's say, if you have the same membrane, but you're gonna do multiple mutations in the protein, different mutations. Different ligands. Oh yeah, then I mean it is possible if you have the same protein, same ligand, same membrane. Yeah, it is possible, but you need to know a lot about how GROMACS works and then write your own scripts and do some coding to, because you can keep the membrane separately fixed and then you can assemble things later on.
I thought that I am the questions. If I change the ligand coordinates,
I mean, if it's little bit of clash is fine. You can, when you minimize, it's gonna resolve. That's fine. That's not a big problem. Yeah, unless there is some ring piercing like this, something like this clashes, then it can resolve. But if it's like just like this, then it can resolve. Yeah, it's not a big problem now. Because you can always do some very careful minimization and relaxation. So for example, you do energy minimization, and then you put some restraints in the protein and slowly release the restraints, the force constant, so it has time to relax.
First, I need to align all this.
Yeah. Yeah, the coordinate is fine. The coordinate is not a problem because it's just PDB, right? But the problem becomes the topology with the connectivity of the ligands. That is the complicated part. Yeah, that's difficult to do automatic way because yeah. You can talk to actually Adam because he's working on that similar thing, like the person sitting in the last, and yeah, his name is Adam. He's gonna, yeah, Adam. Yeah, he's gonna give a. He was talking about some protein ligand high throughput system building. So I think he's gonna talk about tomorrow as well. So we can ask more things to him. Yeah.
You said that
Yeah.
Why do you semi or is the problem to use anisotropic? Because I use gamma T and
Which one? Show me exactly. Just check the GROMACS thing. It's gonna be easier. Now just go GROMACS MDP. Yeah, here. Go here. Which one do you use? Show me. Just the pressure. Just keep going. Yeah.
Finish option.
Yeah, I meant this one. I never used it. Let's see what it says. Yeah, I mean, I don't know. Semi-isotropic because you see, it says that which is isotropic in the X and Y direction, but different in Z. Because in membrane you have the lipids, right? So the membrane itself, it has different properties compared to this one because this is water. So this is lipid, this is water. That's why you wanna have XY is the lipid and Z is the water. So that's why you have this different. That's why semi-isotropic. But here, anisotropic, I think everything is different. So yeah, for membrane, I think this is the recommended one. I never tried this one, and I'm not sure exactly when you would do that as well.
It may be,
and if it's just the solvent, then I just use isotropic. That's the standard one. Why did you use this one? Is there any reason?
I don't know. That's what it is.
Okay.
We are starting with GPSS. Maybe it is like, I don't know.
Unless you find in a different paper where they use it, it is good. I think you should know why you're using this one.
My advisor used this.
Yeah. There might be a good reason to use this. I'm not sure, but I think it's good to at least know why you are using this.
Do you have any
reason?
No. Are you answering any part? Like I'm using 4,000.
Yeah.
Do you use any specific value?
No, it's just standard value.
So semi-isotropic doesn't exist.
No. Yeah. Do you have a question? Okay, okay. Okay, we have just last 5 minutes. You have the results, right? Do you want to post it in, I guess not, maybe everyone doesn't have it, but do you want to post it? Yeah. Just copy paste this and just post the white tape and this to, yeah, to the Slack, yeah.
Karbonlardan zenginler. Öyle reaksiyon mu? Orada diye.
Yeah.
Yaptığım bir şey değil yani.
Sorry? Where? Oh, yeah. Yeah, you need to install MD analysis. Just do pip install MD analysis. MD thread, sorry. Yeah. MD thread. So normally If you get any error, you just copy paste this in the Google and it will tell you what to do. Yeah, yeah.
Everybody has an issue with update, right?
Yeah, I think we're gonna forget about this. Yeah.
Exactly.
Yeah, I'm gonna go to the result now, I think. Yeah.
And after they do analysis, what are the outputs?
Yeah, that thing. I'm gonna do that now. Yeah, yeah. Yes, sure. Okay, great. So yeah. Okay, so all right. So whether you finished or not doesn't matter. So we're gonna go to the results now. Okay. So at the end, so you might struggle with MD analysis, those kind of stuff. You can figure it out later. These are all technical things you can search in Google. But I think the most important thing which you want to learn is that what you can understand about the system, right? That's the whole point of doing simulation. So let's go to that. So this is the system where we asked about, let's say if you open this up, so the idea is that you wanna, let's say, you wanna open this simulation. All right, let's see. Conference, okay. Okay, so this is the simulation. Okay, so this is the simulation how it look like. You start from this long polypeptide, right? It's all alanine. So you can also look at this, like, or other as well, licorice. Oh no, road. This is how the initial system thing was. During simulation, you just saw that it started forming this helix, which is supposed to form. I mean, this is a very small system, so that's why it formed the helix it folded very quickly, but you might not see this folding for anything. Or like for any kind of system, if it's a complex system, it might need to simulate very long time. But you could see that from this one, from this helix here, right? Okay, now we're gonna understand this in terms of what if we do a mutation in this protein. For example, there is one file is called, we mutated one of the alanine to glycine, or we mutated one of the alanine to proline, what happens? So now we're gonna understand this based on this plot. So what we're analyzing here is that with respect to time, this is the time x-axis, the y-axis is the fraction of the number of residues, those are helix here. So as you can see, there is no helix here before because everything is stressed out. It's just the polypeptide, then start forming helix here again. There's one means all the residues are helical. Again, it goes down, it goes up, goes down, goes down, goes down, because it's a simulation. These are all dynamic. So it's not just helix means it's just rigid structure helix all the time. It's gonna sometimes unfold a little bit, sometimes fold a little bit. There's a dynamic part, right? This can happen. As you can see easily here, right? So now you can convert this to a probability because you wanna see now what fraction of residues, how many residues are forming helix around the whole simulation. So how many residues are forming helix here? You see, you can looking at this plot. Look at this number and this probability. Yeah, 11, 12, something like this, right? So this is the highest point you see here. And then now how many residues we have? It's 16, right? Because this is a one, yeah, it's 0 to 15, 16 residues. So out of 16, only 12 are forming helix. Not all the 16 ones. Why do you think so? Exactly, yeah, there's some terminites, exactly, yeah. Because to form helix, what you need is this helical pattern, right? You have to have hydrogen bonds next to each other, right? 1 + 3 or 1 + 4. These are all you have learned about in biochemistry. Here you don't see that because there's nothing beyond it. So you cannot from everything cannot be helix. There's some termini which is not helix. So that's pretty. So that's what you can understand that okay. Yeah. So this is what this plus tells you. Of course there are, and then there's one thing happen is that number 4 there is nothing at all. Why do you think there's nothing in number 4? So if the probability is 0 means not stable, it means unstable, right? Probability is 0, which means that this is quite, it's forming helix for 4 residues very unstable. Why do you think so? No, this is all alanine, right? There is no helix breaker here. Sorry? Yeah, so the idea is that formation of first helix, there's the first helix, right? The 4, you need around 3-4 residues to form it. The first helix is very difficult to form. That's what it's telling you, right? Once it forms the first helix, it forms other helix quite easily. So that is the transition state. That's the energy barrier. Because you can convert this probability to a free energy if you just do Boltzmann inversion, it becomes free energy. This is a high energy barrier, which means that this formation of one helix is difficult because what happens is that if it's a linear polypeptide, it just wants to float around. It has much more entropy, right? And to form helix, you're forming order from this chaos, which means this has to be compensated by enthalpy. That's the free energy, right? Delta is equal to delta. Delta is equal to delta H minus T delta S. So it has to compensate from enthalpy. And the 4 helix is not enough to have the enthalpy. So that's why you are costing the entropy with enthalpy is not enough. That's why there is a barrier here. But once you form this order, then you have much more interactions, much more enthalpy. That's why it's much easier later on to form this helix. Okay. Next, I'm going to go to just one. This is the, which one was it? Proline. Let's put proline. Yeah. So now what do you see here? Now what happened is that there is a mutation in the protein where we mutate one of the alanine in the middle to proline. And what do you see here? What's the difference here? No chance of helix, right? Everything is, yeah, it's just almost 0 everywhere. Why do you think it's happening?
You cannot make hydrogen bonds.
Exactly, yeah, because proline is known as a helix breaker because it has a weird twist. Sorry? Exactly, yeah. Proline has a weird form which is from this weird ring, which means it's difficult to form hydrogen bonds between the residues neighboring.
There's a difference between proline and alanine.
Yes.
May I interrupt? Some people need to be.
Sorry? Oh, some people need to leave, yeah. Okay, all right. That's the idea. So proline is a helix breaker. That's why we don't form helix. That's it. Yeah, yeah, yeah.
Okay. Thank you so much.
Yeah. Yeah, yeah. Yeah, I hope, yeah, everyone liked it.